on fish biology and ecology. But before we start with this session, I'd just like to mention a few things. And one of them is that I don't know if you were aware of this, but today is International One Health Day. So those of you who don't really know or are comfortable with the One Health concept might ask, why do I mention that when we talk about fish biology and ecology? Now, the One Health concept was formulated as an interdisciplinary approach to achieve optimal health for people, animals, and the environment via collaborative action across health and environmental sciences. So the thing about the One Health is that it was originally formulated by vets, and then the medical fraternity really quickly bought into this. But the one important aspect of the triad that hasn't really received as much attention that it should have had to date is the environment. And a big important part of the environment is freshwater and our dependence on freshwater and freshwater health. So when we start to talk about fish and the role that fish play within ecosystem health and how it can be used as a biomarker of health and how important freshwater fish are to indicate healthy systems, then I'm sure you should start to agree that knowing about the ecology of fish, the health of fish, the diversity of fish actually do play a part in the One Health approach. And I don't think anyone in the world can now argue how important it is that we have this integrated approach to look at ecosystem, human and animal health, just due to the whole coronavirus that we're still busy battling with. So I'm looking forward to this session. It's a lot of 12 different presentations, really great presentations. I hope you had an opportunity to go through the presentations, the abstracts, as well as the presentations themselves, and that from that we can have a really great discussion. So what I've done is I had a look at the 12 different presentations, and then the main themes that I can see from that is, first of all, the ecology of the fishes, where we look at different ways of how you determine the ecology. Then there's quite a grouping of presentations on taxonomy and the phylogeny and diversity of fishes. And then the third one on the ecosystem health. So that's how we can use the fish in different ways to determine ecosystem health. And then that speaks to the whole One Health concept more directly as well. So what I'm going to do for this session is that I think we take it in little bite sizes. So we first start with the ecology of fishes, and then I'm going to ask those the presenters that presented work related to the ecology to just introduce themselves and give a 2-minute, 3-minute highlights of their presentation. And then following that, I will open the floor for discussions regarding the work that has been done on the ecology. What I would like the authors to do, if you do your little chat, and if it's possible for you, just switch on your camera so that we can have an experience of seeing a live person in front of us, just for those few minutes. So what I have for the ecology section is we've got Casey Broom looking at the prey type and determine influence functional responses of threatening Cape Fold ecoregion fishes. Then also Anna talking about the distribution and habitat association of freshwater fishes. Then also Matthew Burnett, you did a nice presentation on the temporal spatial ecology of Labiobarbus species. And then the final one for this section of this what we're now busy with is using stable isotope analysis to look at the trophic ecology of the fishes as well. And that one was by Tholoane Lona. So please, if those authors that I've just mentioned, if you can just give us a quick introduction to yourself as well as the work that you've presented, and then we can discuss it. So Casey, if you're around, if you can just open your video and have a chat with us. Thank you.
Hi, thanks, Nico. Can you hear me?
Perfect.
Hi. Sorry, I unfortunately don't have access to a webcam right now, so I can't turn on my camera. Anyway, I'll just introduce myself. So, hi everyone, I'm Casey. I'm completing my PhD at SASAqS through the Department of Ichthyology and Fisheries Science at Rhodes University. So this presentation was based on an experimental component of my PhD, which has also been published in Environmental Biology of Fishes. So it links to my wider thesis looking at the recovering Rondegat River cyprinid assemblage, which also investigates functional morphology, diet, habitat use, and behavior of these fishes. And so in this experimental study, we looked at the Clanwilliam redfin and the Clanwilliam yellowfish, which co-occur over the majority of the river. We wanted to compare their feeding performance using functional responses to better understand their trophic interactions and competition in these species. So to do this, we determined the feeding rates and the functional responses of these two species in the lab by feeding them on mosquito and chronomid larvae at different field representative temperatures of 18 degrees and 25 degrees. And so we expected that the redfin would outcompete the yellowfish in the surface and water column prey, which was the mosquito larvae, while the yellowfish we would expect to be more efficient at feeding on the substrate-dependent chironomids. And in fact, we found that the yellowfish outperformed the redfin on both prey types and at both temperature treatments at higher prey densities. The redfin, though, did have an initial high attack rate, especially at the lower temperatures, so it was more able to exploit prey at low densities. And then the high performance of the yellowfish was mostly driven by shorter handling times, especially on the chironomids, compared to the redfin, which had quite a varying handling time on these different prey. And then on the temperature side of things, they had a few different effects on prey consumption dependent on the fish species and prey type. However, they were not significantly different in their functional response between these temperature conditions for either the prey species. And so we took this to indicate some level of community resilience to thermal change in the Cape Fold ecoregion. And that's about it for my study. Thank you.
Okay, great. Thank you very much for that summary. If we move on to Anna Ntombeni, your work on the Crom River.
Thank you, everyone. Greetings to you all. Thank you for this opportunity. Can you hear me all?
Yes, perfect.
Okay, just a minor change on my topic there on the program. My talk will only focus on the distribution of the freshwater fishes in the Crom River. So to take from what my supervisor, Dr. Albert Tagona's presentation, my work is an ongoing work whereby this ongoing work has highlighted a discovery of new species and lineages. And some of the species that were thought to be widely distributed are actually narrow-range endemics, but they occur in highly modified environments. However, our knowledge on their distribution patterns is lacking, and it is essential to understand where these threatened species are distributed. Therefore, my talk presents the distribution range maps based on the historic and present distribution of the fishes in the Crom River. And from the results, we found that the native species have now moved towards the upper sections of the river, and the localities that the native species were found historically are now invaded by the introduced alien fish species. The current distribution of this native freshwater taxa has decreased as compared to the historical distribution, and this is alarming because the results suggest that some of the threatened species might have now been already driven to extinction, and the critically endangered species are now highly impacted. And should the Crom River system not be prioritized and conservation measures taken, this remnant population of the native species will be negatively impacted or even worse, driven to extinction. Therefore, the study would like to recommend that the long-term conservation measures focus on preventing the spread of these non-native species and rehabilitate the critical habitat for future persistence of this native population. Thank you.
Thank you very much for that summary. So we go on to Matthew Bennett, if you can tell us about your work on Lobia barbus.
Morning, Prof. Nico, and good to be at SASAqS today. Yeah, and thanks for the opportunity to share some of the work that we've been involved in here in KwaZulu-Natal on the Labiobarbus species, particularly Natalensis. And what we did is we used radiotelemetry techniques that were locally developed here in South Africa to track the movements, both the temporal and spatial movements within the Mgeni catchment, and with some of the responses to flows and temperature. So some of the study came out, we found that temperatures cued behavioral movements, but flows actually provided access to fish to get to the habitats they needed. What was really interesting, some of our previous work is we found that yellowfish are facultative migrators, meaning that they will move between habitats based on where those habitats are, and less so on longitudinal movements. But what we found with the yellowfish in the scaly yellowfish is that their movements are quite distinct over seasonal patterns between deep pools for refugia habitat in the winter, and then these riffles and rapids in the river habitat for spawning during the summer months. So that's just some of the findings that we had around the work that we were doing. I think also what was important is we're trying to link these to setting and making environmental flow requirements for managers in the system, and kind of showing that higher base flows during the summer are probably more important and need to be considered so that the yellowfish can access spawning sites during the summer months. So I think what lastly on that was there were some in-stream barriers that were that did obstruct the movement of fish, although fish could get over them, and that does hinder the fish movement, particularly when your environmental flows aren't met, and it restricts the access to habitat from when they move between these refugia habitats and the spawning grounds. So yeah, thank you.
Great, thank you, Matthew. So for the final presentation in this little series, we're going to go to Luana. If you're there, to just tell us about your isotope work that you did.
Good day. Good day, everyone. I'm Cuomo, an MSc student from Rhodes University. Sandela benza, which is my study species, is an endangered species from the Eastern Cape province. A recent study done by Chakona et al. Found that there are actually 3 divergent lineages within Sandela benza, which are allopatrically distributed in the river systems of the Eastern Cape. Because the species is listed as endangered by the IUCN, this raised some concerns that these lineages could be facing a higher risk of extinction, as they may potentially have narrower geographical ranges than currently recognized. Therefore, it is important that we integrate genetic, morphological, ecological, and other biological characteristics in order to determine the taxonomic status of the lineages identified within Sandela benza. This study therefore looks into the ecological aspect. We would like to see how different or similar the systems occupied by the 3 Sandela benza lineages are in terms of their food web ecology using stable isotope analyses, as well as whether the isotopic niche patterns of the lineages vary in their respective communities, and look into the interspecific interactions of Sandela benza lineages with other co-occurring species. The results of the study showed that the river systems occupied by the different lineages varied in their food web characteristics, and that the lineages differed in their isotopic niche patterns and trophic interrelationships in their respective systems. These findings will help in accurately resolving them as new species, as well as assist the researchers, conservationists, amongst others, in sustainable partnership programs that will promote improved management practices for sustainable functioning of rivers and to protect critical habitats for these newly discovered lineages. Thank you.
Thank you very much, Sulusile, for that summary of your work. So I'm gonna open the floor now for questions to any one of these four authors on their work on the ecology or related to the ecology for fishes. And just to start off the discussion, Casey, I would like to ask you a question. I saw within your presentation what you rightly so did is you made sure that the two different species, the fishes that you compared, were of similar size. But what I was wondering about is that we know that the Glen William yellow is much bigger, or it grows much bigger. Did you maybe think about the age of the fish that you compared to one another? Because surely the Glen William yellow must have been a juvenile compared to a more adult of the other species. So just your comments on that, please.
Thanks, Nico. Yeah, you are very correct that these would have been juvenile yellowfish, and we tried to size match them as close as possible. So we needed to have, you could either compare them at different sizes, and just in terms of keeping things as comparable as possible, that was something would have to give in the equation, obviously. But I think the main reason why we decided to go this way was that from our observations on the river, we have these large aggregations of juveniles in a much higher abundance than the larger adults in the river, and so they are ecologically more relevant as a competitor and relevant to the interactions between them in terms of the sheer numbers of fish interacting, while you have relatively fewer large adults. So I think the comparison is potentially more useful this way around, but it is a good thing to keep in mind. Thanks for that.
Okay, great, thank you. Then I see we've got Wynand Vlok. You've got your hand raised for a question.
Thank you, Nico. Good morning to you and to the rest of the people. Casey, yeah, I think my question is similarly to Nico's, and I think you've partially answered it, because I think it's also important that you are looking at the size class, and there might be feeding preference change as they go a little bit bigger. And then I think something that one must also maybe take into consideration, especially if they go into just a little bit of a larger class size, is the importance of water depth and water column, because I think then there will be a differentiation. But I think your interpretation of using the smaller or the similar groups at a younger age was based on what was observed in the river. So thank you very much for that.
Thanks, Van. Yeah, so I've also been looking at the functional morphology and behavior and diet of these species, and you're very correct that there is that trophic shift as they age, and that's an important one that we might need to look into in more detail, because obviously some of the diet data and I'm hoping to get trophic, stable results back fairly soon. A lot of this will be based on the adults, but we also specifically got samples from juveniles and from adults and separated them in the data sets. So hopefully that'll be able to differentiate that a little bit and give us some more insights from the other chapters in the PhD towards this. But this is just a small part of the puzzle so far. But yeah, thank you for that.
Thank you, Casey. Then Leon Barkhuizen, you've got your hand up for a question.
Thank you. My question is to Matthew. I think you will be aware that similar studies have been done in the Vaal River system on Labiobarbus campbellensis and Labiobarbus aeneus. How does your results of Labiobarbus natalensis compare to those studies that was done in the Vaal River?
Yeah, thanks, Leon. So that was most of the work done through Dr. O'Brien's work and some of my undergraduates in MSc. And that was, I tried to allude to that a little bit, is that generally we find that the yellowfish move between habitats required that they need for either foraging or refugia or spawning. And the spatial kind of distribution of these habitats varies within each ecosystem, so they don't necessarily need to move long distances, as long as that these habitats are within, or as long as they can reach these habitats. And so with some of the work that we did in the Crocodile River with Labiobarbus moraquensis, was that they didn't need to move further than 1 kilometer because they had the habitat requirements that they needed within that kilometer stretch of river. That's similarly to what we found on the Vaal River system, although we also know with doing these behavioral studies, looking at individual movements, is that you often get a flyer that one fish will decide to move over 10 plus kilometers. And for the majority of my study here on the Mgeni catchment, was most of them were pretty, showed high site fidelity, meaning that they didn't move much, except with the exception of one that moved the entire reach that we were studying. With the Labiobarbuses on the Vaal system, I don't think with those studies we actually tagged them over a long enough period to detect some seasonal movement in and out of this of the study area. So that's also something with this study, we tracked them for a full year period, and we could get a good seasonal kind of variation in their spatial movements. And so as we continue to do this work, we're actually developing it and getting kind of more fine scale movements for these fish species. So I hope that answers your question. The one other aspect, what we found was with temperature cues is very important. And I'm not so sure much on the Vaal because that wasn't really unpacked much in those studies, but with this study, we definitely shifts in temperature of the river was cueing the behavior, the movement of the fish in the system.
Thank you, Matthew. While we have you on the line, I also have a question for you. In your abstract, you mentioned there part of the motivation of your study is to understand how the species has adapted to these anthropogenic changes. How did you apply your results to actually understand the adaptation of the species to the changes as you put down in your abstract?
Thanks, Nico. I think it's very hard because we didn't have kind of pre-anthropogenic conditions to look at. But what we did look at was the kind of construction of in-stream barriers and how they moved over those. And what we looked at was when fish were moving. And what we found is when flows were low, fish would largely move at night over obstacles. So a lot of the upstream movement happened when the stream flow was still very low, and a lot of the time the fish would then move at night to do those movements. During the higher period of our study, for fish moving back into this their refugia habitat, the flows were elevated and had good flows, and movement then happened primarily during the day. So we assume that to adapt to these changes, they are moving at different times of the day, and so they're not exerting that deal behavioral pattern that they're known to have.
Okay, great. Yeah, no, that helps a little bit. But you're correct. You don't have historic data, so then it's difficult to try really to understand how they adapt. So Wynand Vlok, I see you've got your hand raised.
Yes, thanks, Nico. Hi, Matthew. On the temperature issue, I think it is very important. One of the comments you've made, and maybe I've missed it, but you talked about the flood releases and maintaining ecological flows. I think what is important is that one must also remember that with many of the impoundments, especially the older ones, releases are normally bottom releases, which will have poor water quality and the water will be much colder. I think if we remember back what happened in the Clan William system, when that was happening with the cold releases from that impoundment. So I think that is also when one make these recommendations, I think for a system to maintain the populations and spawning activities, it will be important that must be taken into consideration, especially if one is looking to use the cue of temperature for spawning and migrations. Thank you.
Thanks, Wynand. If I can just comment on that, Nico.
Yes, any comment, welcome.
So that's very true, Wynand. And what we had is we had temperature loggers throughout our study area. I think for the spatial movement, we just highlighted 2 of them to draw the comparisons and the cues that were really in close relationship to where the fish were. But we did have a probe below Mid-Mar Dam. And what we found very interesting is that there was some remedial effect of the temperature release after the bottom release from Mid-Mar Dam. And the temperature difference between downstream and upstream of that release was close to about 10-15 degrees Celsius. So the bottom releases from Mid-Mar Dam were a lot colder, but we saw that it returned to kind of normal environmental temperatures after Howick Falls waterfall, and into the system where the yellowfish were primarily congregating around. So you're definitely right, it needs to be considered, but for this particular study, we actually found that there was remedial effect from a bottom release on Mid-Mar Dam. I think with that though, and we're still looking at some of the stuff, is that we find that these environmental releases, which we did one during the study, was they do top releases for the environmental flood release, but they release it over maybe at most a 48-hour period. And even though they try stagger it to mimic a kind of consistent increase, they drop it off very quickly. And we're finding that even though they do a flood release, they drop the base flows go from 2 cubic meters per second up to 20 cubic meters per second, and then back down to 2 cubic meters per second because they don't want to, release more than what's necessary. And we're finding that those flows are actually probably as disruptive to the fish movements as whether they would rather just maintain a higher base flow during the high flow season. But we're still unpacking that a little bit with the data that we have. So thanks.
Great, thank you, Matthew. I think for the ecology part of this discussion, Gordon wants to make a comment. Gordon, if you can make a quick comment, and then we move on to the 8 presentations regarding taxonomy and systematics. Thank you, Gordon.
Hey everyone. Great to hear your voices. I wish we could see each other in person. I just wanted to add to this discussion about fish behavior. And one of the big comments that we get internationally is that you're altering its environment, you're putting it under stress, you're attaching this monstrosity of a tag to the fish, and how can you expect it to give you an indication of its natural behavior and use that to describe its biology? So this is where this adaptability issue comes in. And I know that we skirt this issue a lot, but we do, and we need to acknowledge that we work in altered environments and altered conditions because of what we're doing to the fish when we undertaking these studies. But what we rely on is that the fish, in order for it to survive, and the long-term experiments are very useful because at least we know that the fish is able to survive and sometimes recruit. It has to then carry out and perform some aspect of its natural biology and ecology. And that's kind of where this argument is going, is that we don't hide away from the fact that we're changing its conditions and the environments that we're carrying these tests out. But what we know is that they survive and they recruit and they feed. And how do they do that? And when do they do that? And what are they doing? And we can use that to hypothesize what use that to provide a hypothesis of their ecology or biology. And that's where we are at the moment. Thanks.
Thank you for that, Gordon. So please, if you have any other questions regarding this to the different presenters, put it down in the chat. Then everyone can see the questions and the answers as well. So presenters, you can answer in the chat in the meantime as well. If we have time at the end of the session and there's more discussion on the ecology, we'll come back to that. Okay, so then a very nice big part of this session was devoted to taxonomy, systematics, and diversity. And I think that fits very nicely with Albert's presentation this morning. So if those authors, if you can just introduce yourself quickly and give a short, brief summary of your work. Pedro, we can start with you and your work on the lamp eyes.
Hello, good morning. My name is Pedro. I'm a postdoc at SASAqS, and my talk will be about a group called African lamp eyes. They are like small fishes occurring in freshwater environments. And I will make an update about the taxonomy of the family in general, and more specifically about the Lacustricola genus in Southern Africa. Talking about my research since I finished my PhD, going now to my recent research as a side postdoc in the last 2 years, where we describe new species and we try to, strategize on how to deal with a group that have a lot of diversity, little known knowledge about the taxonomy, like species that were described really like 100 years ago with brief descriptions. So I'm going to talk about that, how we can study taxonomy. That's the base for all other studies in a group that is that difficult, complex group that is difficult to delimit species boundaries, how we can do that. So yeah, that will be my talk. Thank you.
Great, thank you very much for that. Martinez, your work on the barbs.
Good morning, can you hear me?
Yes, we can.
Yeah, I'm Martinez. I'm a PhD student at SASAqS and the Geology Department at Rhodes. And my talk was about starting to understand the phylogeny of South African barbs, of mostly the genus Interomius. It's got a lot of diversity. There's about 220 something species distributed through Africa, and about 45-ish in Southern Africa. So we used CO1 bulk to, first of all, get an idea of the systematics and the relationships. And then we also wanted to test Paul Skelton's groupings of the barbs into soft-fin barbs, fine-fin barbs, and soft-ray barbs to see whether it forms natural groupings. And so we recovered 3 major clades, one being largely central to Western Africa, and the other 2 largely Eastern and Southern African clades. And then the groupings into different soft-fin, spine-fin, and soft-rayed barbs did not turn out to be natural groupings. So we're still trying to figure out what connects these clades together. And then to move forward, we need some more taxon sampling. We stick to specimens that we could sequence from their type localities so that we don't get messed up with misidentifications and range distribution problems. Some of these species have got massive ranges, and I've had a quick couple of looks at some species, and they do seem to form different groups based on the river system. So we need to look at that in the future and sort that type of thing out. And that is the main things that was in my talk. Thanks.
Thank you. Thank you very much for that. Yunela, your work on Parakinoglonis.
Good day, everyone. I'm Yunela Stolle. I'm a PhD student at SASAqS and Rose University. So in our study, we used integrative taxonomic approach combining color pattern, morphological, and molecular data to describe Parakinoglonis specimens that were recently collected from the Lubudi River in the Congo Basin as a new species to science. So to do this, 78 specimens of Parakinoglonis from 6 Parakinoglonis species in the Congo Basin were examined. Some of these species were differentiated into sub-basins based on color pattern. And our results, we show that the Lubudi specimens are distinguished from all Parakinoglonis species occurring in the Congo Basin by bars on the side of the body, while all other species are spotted. These specimens also have broad humeral process embedded under the skin, while all other species have a narrow humeral process that is visible through the skin. The anterior margin of the pectoral fin spine is mostly serrated, while in other species, it is mostly smooth. The Lubudi specimens were also further differentiated by combination of morphometric characters from each species or populations in the Congo Basin. In the genetic analysis, the Lubudi specimens formed a distinct clade with a 94% bootstrap and genetic distance of more than 3%. Therefore, based on color pattern, morphology, and genetic evidence, these specimens are currently under revision, under description as a new species. Furthermore, our results also show that or highlighted the need for further taxonomic revision for other Parakinoglonis species, such as Parakinoglonis ngamensis, which is the only Parakinoglonis species occurring in Southern Africa. It showed morphological differences with the populations occurring in the Congo Basin. Another species that is showing genetic differentiation was Parakinoglonis punctatus. And the fifth species that is said to be occurring in Congo Basin, that is Parakinoglonis pantherinus. In our study, we confirmed that this species is a lower Guinea species and it's not occurring in the Congo Basin. The population in the jar is yet another new species. Thank you.
Well, that's very exciting, especially having another new species in the jar. That's great stuff. So next, we've got Thuloana on your the work that you did on the taxonomic diversity of the Zambesian low-veld ecoregion.
Hello, my name is Thuloana Dukwane. I'm a student at the South African Biodiversity Institute at SASAqS. So this study was part of my PhD study on freshwater fish taxonomy. So for this chapter, we did a literature review on the diversity and distribution of the Zambesian low-veld freshwater ecoregion. So this region has the highest freshwater fishes in Southern Africa. However, the information on the fish diversity in this region has yet to be comprehensively documented. So in our review, we found that a majority of these fishes are listed as least concern because of having wide distribution ranges. But recent studies on the fishes found in the river system of the ZLE are actually found to be species complexes, and subsequently the species described were narrow endemics. The second result we could ascertain from the study is that there were river sections which were completely left out in any sampling event. So there hadn't been any people who had sampled in those regions. So this information is important to guide efforts to fill the gaps in the diversity of these fishes, and the better knowledge in the diversity will allow for identification of biodiversity hotspots for conservation, which will allow resource utilization without destroying the biodiversity, because part of conservation is making sure that humans also benefit. So more knowledge will help us to achieve every goal we set out. Thank you.
Wow, thank you for that really interesting work and showing the incredible diversity that we have. So then on next to the Diwa, not one, but two, but 5 new species of momirids.
Good morning, everyone. Can you hear me?
Yes.
Okay, so thank you very much for this opportunity to share with you some of the work that I've been doing. So I've been working on the momirids, better known as the elephant fishes. It's an interesting group of fishes that are particularly well known for their ability to use electric organ discharges for communication as well as exploring their environment. So for my study, I was particularly looking at the Hippopotamus ansouji species complex. So currently, as it is described, it has a very wide distribution, which stretches all the way from Angola in the Kwanza River system, all the way down to the eastern Zimbabwean islands in the Buzi River system. This is a particularly wide distribution. But within this distribution, some studies have shown that there is some diversity within this group, but no new species had been described from this species complex. So one of the major impediments towards the description of new species in this group, despite there being some knowledge of some diversity, was that there wasn't much information concerning its type locality, which was given in a very broad range, very broad area. So we didn't know the type locality. So in terms of describing new species, that was one of the major challenges. But with my work, we actually were able to synthesize some of the information and we were able to track down what we proposed as the Kwanza is the possible type locality. And this enabled us to then go and then address the new species, the distinct groups that we knew were in this group without the risk of redescribing something that was already described. So in my study, we looked at the genetics and we were actually able to find 12 molecular taxonomic units throughout the range of Hippopotamus ansouji. And from these 12 molecular taxonomic units, we were able to describe 5 new species based on both the genetics as well as the morphological data that we presented. The other species, molecular taxonomic units, were not described in the study because, while some of them are actually related to some currently valid species, and there is ongoing work to actually then trying to link some of these lineages to the current species. But the 5 new species I described were mostly from the eastern side. We had 3 species from the eastern Zimbabwean islands. We had 2, one from the Pungwe, one from the Buzi, one from the Ruo. And then we had a species, one from the Kwanza and one from the Okavango River system. So these were allopatrically distributed, so there was no risk of redescription of currently existing species. And all of them had distinct morphological characters. So in essence, my study just highlights that there is a lot of currently, as Albert has already mentioned, that there's a lot of diversity that's hidden within Southern African freshwater fishes. And there is a lot of work that we still need to do in order to currently understand the diversity as well as to provide more precise distribution patterns for these species. Because as it is, ansouji is classified as least concern, but from my work, splitting it into 12 different groups, you realize that most of these are very narrow ranges that are quite may fall within the threatened categories on the IUCN. So yeah, that was the gist of my work. Thank you.
Well, thank you very much for that. Then our last speaker in the taxonomy diversity part of this session, Manda, is going back to Enteromius. We've heard about the complexity within this group. So can you just can you just tell us about your project?
Good morning, everybody. My presentation is on a recently published paper from my PhD work titled Allopatric Differentiation in the Enteromius Anoplast Complex in South Africa, with the revalidation of Enteromius cenuis and Enteromius oraniensis and the description of a new species, Enteromius mandelae. So the Charlie-head barb Enteromius anoplast was the most widely distributed freshwater fish in South Africa, occurring in almost all the major river systems. And the use of a comprehensive data set of mitochondrial cytochrome B sequences uncovered the presence of 4 distinct lineages within Enteromeres anoplus. The Huitz lineage endemic to the Huitz River system, the Olifants lineage endemic to the Olifants-Dorian River system, the Orange lineage endemic to the Orange River system, and the Eastern Cape lineage, which is distributed across 6 river systems from the Great Fish to the Buffalo Rivers in the Eastern Cape province. So the study also provided a new diagnosis for Enteromeres anoplus sensu strictu from the Huitz River system. It also revalidated Enteromeres senus from the Olifants River system and Enteromeres oranensis from the Orange River system as distinct species. The study also provided a description of a new species, Enteromeres mandelae, from the Eastern Cape province. So Enteromeres senus and Enteromeres mandelae could be differentiated from Enteromeres anoplus and Enteromeres oranensis by having a complete lateral line, and Enteromeres senus could be further differentiated from the other 3 species by having long maxillary barbels. Enteromeres oranensis was found to be the other 3 species by having inconspicuous barbels. So yeah, that's it. Thank you.
Thank you very much. So that's all our speakers with taxonomy, systematic and diversity. So if there's any specific questions for them, if you can put it in the chat or raise your hand, and then we can have a look at it. I can just say from my side as an aquatic parasitologist, also involved in species descriptions, it's always great to find new species, but the fishy guys describing new hosts for my parasites sometimes make it a little bit more confusing, but it's great that we can resolve it and hopefully resolve all those interactions together. I don't see any hands raised, so I'll kick off the questions. First of all, to Diwa, what I would like to ask you is, I know that a few years ago, researchers from University of Johannesburg and Germany actually described new species of momyrids, a lot based on the difference in the signal of the electric impulses that they produce. Did you maybe consider looking at that as well for your species, or was that only based on morphology and molecular work?
Thank you very much, Prof. Nico. Yeah, that is correct. I think you're referring to a lot of work that has been done by Bernd Kramer, I think, and his group. Yes, for the momyrids, the electric impulses are actually a very important taxonomic character that has been used in a lot of groups. But for my study, I wasn't able to look at the electric impulses for some of them because we're working with specimens that have been collected over a long period across multiple expeditions by different people. So I didn't have the data for some of that, but we do have electric impulses for some of the species, particularly from the Eastern Zimbabwean Islands, which were collected more recently, and some within the Okavango. So I didn't include it in this paper, but it is a very important character. And hopefully, if we do get an opportunity to go to some of these locations, which are actually very remote, like the ones in Angola, we might be able to look at some of these electric organ discharges for some of them. But yeah, in short, we have some information for some groups, but we don't have for others. But from the genetics and the morphology, we also have characters that we can use to distinguish the different species. So yeah, it will be a nice extra layer of evidence, but for now, we also have enough. Thank you.
Okay, great. Thank you. I think you've got their motivation for some really cool field trips to go and find those live ones. I see Leon Barkhuizen, you've just raised your hand.
Thanks, Nico. I want to refer my question to Manda and Martinez. Did anybody look at the Enteromeres species in the Orange Vaal River system? Because historic data of fish surveys done in the 1970s indicate that Enteromeres pallidus occur here. And yes, sometimes we find minnows and we are struggling with the exact identification. So if I can maybe just tell us more about, has the work been done in the Free State and Orange Vaal River system specifically? Thank you.
Okay, so it was addressed to two authors. Maybe Martinez, you can start, and then Manda.
Yeah, I think Manda might be better for that one. Most of her work was in the Orange and Western Cape region. I think she'd probably be better to help you there.
Yeah, the work we didn't really look at the northeastern portions of South Africa, so that's what's next on the list. So that work is basically pending to see what exactly is in those river systems, if it is Enteromias anoplus and what other anoplus species are found in there.
Okay, so there's Leon, there's some nice future work collaboration for you there, getting some samples down to SASAqS so that they can resolve that for us as well. Matthew Bennett, you've got your hand up.
Thanks, Nico. Yes, on with the Enteromia species and Anoplus, we've got reports of guys transferring them into trout waters to feed trout. Was with the genetic makeup and was that considered in the study, and was there any kind of historical data to try and look at tracing the genetic strains with introductions from people around the trout industry?
Okay.
Sorry, I don't know, was that my question?
That's okay, you can comment on it, that's perfectly fine.
Well, from my study from the 4 Enteromia species, we found there wasn't much genetic mixing except from the Enteromias mandelae from the Eastern Cape. So there was mixing in some of the river systems from the Buffalo, Keiskamma. So we suspect that is due to, as you've said, trout fishing. So that was the only mixing we found. Otherwise, they're pretty genetically distinct.
Thank you. Byron Grant, you've got an up.
Thanks, Nico. Just to respond or provide a little insight for Leon, Albert's group did do a review of the Palladus species in 2015, and they also produced an article over there, and they did confirm that the Palladus in the Vaal system was genetically distinct from the type location of the Palladus in the Eastern Cape. And I do believe that they are planning collection trips probably in the next year to try to get to some of the localities of the Palladus group to try to resolve some of the taxonomy on that. So yeah, I think that work is going to be coming up, and they have done preliminary work on it already.
Byron, thanks for that. Valant Malherbe.
Okay, so there's questions that came through the chat and some of the YouTube videos. The first one is from Peter Kotze to Manda Kambikambi on, do you have any idea where the Enteromias Anoplus from the Anoplus from the Limpopo, Olifants East and Inkamati fit in? Completely different lineages or species is the question.
Yes, we do suspect it's completely different. It's completely different from Enteromias Anoplus, but we will have to verify that with some genetic and morphological work. So that's still pending, basically.
Great, thank you. Valant, you're going to just run through all of them, all the questions.
So there's another question for Sifu Sake Mgluli on, you mentioned in your talk that the male fish grew faster than the females. Do you know what the reason for that was? Not sure if, do you hear that question? Not sure if the presenters, yeah. And then there's another question for Nobuhle Mpanza, I think that's okay. wrong session, I think. So that's all questions for now.
Okay, thank you, Fainan, there from the organizer's office. So I think we can almost wrap up this part of it. I just want one more question. I think I'm going to address it to the Martinez, but if any of the if Albert is out there and the people doing all the lovely genetic work, they can maybe pitch in as well. I see, Martinez, your focus for your phylogeny was on CO1, mitochondrial gene. Do you think you'll get the same results if you include nuclear genes as well? I was really pleased to see that the Diwa actually had a look at nuclear as well as mitochondrial genes. So I know with the work that we do on invertebrates and parasites, we have to do at least 3-4 different genes. We have to include nuclear as well as mitochondrial genes, especially nuclear genes if we do higher phylogeny. I'm not sure what is the case within fish, so I'd like to hear your comments on that, and anyone else would like to pitch in.
Yeah, I think Albert has gone fishing again. I would ideally like to add some nuclear genes to this. I think the CO1 might not resolve your very deep nodes that well. So I think my topologies all remain quite similar, no matter what methods I use. So I think it's fairly robust at this stage, but like I said, I think nuclear genes, adding a few of them would be nice. And there was 2 studies came up a few years ago that the one only used site B, and the other one included a couple of nuclear genes. And my main clades are quite similar to these. So I think for now that gives us a good indication of what's going on. But I think adding more taxa and loci will improve the resolution of the tree, if that answers your question.
Okay, great. No, it definitely does, and I think it's once you've extracted the DNA, it's not that too difficult to add the different primers of the other genes and get those results in as well. So great work and keep it going.
Thanks.
So we'll get we move on now to the final presentations in this session, and I've grouped them under ecosystem health. And again, just if you have questions still for the other speakers, please put it down in the chat, and they can answer it directly, or we'll get back to that. So first of all, Annelies, if we can go to you and you can tell us about the state of the fish communities in the Nkomati catchment. Thank you.
Hi everyone. So I'm Annelies, a master's student finishing up now at the University of Mpumalanga. My study was about assessing the fish communities in the Nkomati and how the different environmental drivers affect them. So I sampled the Sabi, Crocodile, and Nkomati rivers and all the fish communities within. We found that within this whole Nkomati catchment, mostly all of the environmental drivers had significant influences on the fish communities. These included the water quality, substrate, velocity, depth, cover features available to the fish, and all of that. So for my study, I mostly identified that velocity and substrate are the main drivers that should be prioritized for managing and monitoring, as these drivers, if the loss in these drivers will affect the fish communities the greatest or influences them the most. So this is mostly due to all the barriers we have in the system or the catchment and the main issues we have with sedimentation, input into the river systems, and sand mining, and all of that. So yeah, that's just a quick broad summary of, yes, up one chapter of my master's.
Great, thank you, Annelies. And then the final speaker in this session, SASAqS, if you're going to tell us about your histopathology-based self-assessment of Clarius Grippinus.
Hello, everyone. Can you hear me?
Yes, we can.
Okay, my name is SASAqS, and I'm a master's student at the University of KwaZulu-Natal in the School of Life Sciences. So this talk today is one part from my master's research project. So Umgeni River is an important river providing water to about 5 million people in Durban and Pietermaritzburg. And it's very important in the province economy because it contributes about 60% to the province GDP. Yet it is mostly polluted, and recent studies have highlighted that the river is receiving huge pollution loads from different land uses. However, there are no fish health studies that have been conducted in the system. So this study used Clarius Grippinus as a sentinel species to see how it will respond to the level of pollutions in the Umgeni River. And we followed the use of biometric indices and the histology assessment. And the results have shown that for the water quality, it is deteriorating more in particular in the Inanda Dam. And both the dams have recorded mesotrophic conditions for the nutrients, and there were low concentrations of metals in water as compared to the sediments. The biometric indices show that fish was in a good condition, which is can be attributed to the fact that Clarius Grippinus is an omnivorous and opportunistic feeder, so it basically feeds on anything that's available. And the results from the histology have shown the alterations such as aneurysm, hemorrhages, hyperplasia, leukocyte infiltrations, the MMCs, epithelial lifting in the gills. And these alterations were more prevalent in populations from Inanda as compared to the Nego Dam. This goes for the other organs, the liver and the testes and ovaries. Despite variations in histological alterations in both populations, the severity of alterations were categorized as slight to moderate. However, the effects of pollution in the Umgeni River is still a cause for concern. Thank you.
Thank you very much for that. So we've got now the floor open for questions for these two presenters on ecosystem health. I can open the floor with a question to Annelies. You mentioned there that you got 39 of the 53 expected species, so you were missing 14. What would you say, was that down to not being able to catch them using the different techniques, or you think they're absent or very scarce? And what can you take from those missing 14 species?
Hi, Niku. Thanks for the question. Yeah, I guess it was just the timing we went sampling and the effort we've put in, because a few surveys after which I did my data collection, I've actually caught more species in other events, which I didn't caught while collecting for my master's. Yeah, and then mostly it's like CTR brevis, which is also critically endangered, which I didn't find during my surveys. We caught it then after my surveys, and Cynodontis, not Cynodontis, Serranochromis meridianus, we also found in the Sabi, and tigerfish we found in the Sabi, but yeah, these were absent from when I did my collections. So I guess it's just effort, and then also the timing of sampling from my side.
Okay, thank you. I also see Vainant have a question. There's in the chat box linked to one that I just asked. So he said that some clarity, he would like some clarity, was the vegetation, especially overhanging and in-stream, was that in a good condition? Because smaller fish prefer these biotypes, yet at lower flows, vegetation cover is absent, especially related to flow variation and drought scenarios. So comments on the vegetation during your sampling period?
Hi, Bainan, thanks for the question. So the vegetation in the riparian zone and stuff weren't too bad, but we found that a lot of the sites had invasive reeds or different invasive plants that that grew there. So I'm not sure if it can be the absence of fish can be attributed to that. That's a good thing to look into. But yeah, low flows were mostly, the banks were high and eroded away, so no vegetation in sense of overhanging was available for the fish to utilize.
Thank you for that, Matthew. Vanette, we've got a question from you.
Thanks, Nico. Hi, Annalise. Thanks for your talk. I just want to ask about your reference species list to in relation to what you expected to find and what you did find. Similarly, in some of the studies we've done in KZN and with some of the taxonomic studies that are coming in, is that we're looking at species distribution across certain ranges, and we actually have very little detection of these kind of more cryptic species that we expect to find, but when we actually go out looking and doing these community assessments, we don't find. So yeah, just maybe a comment on where you got your reference list to find out what you kind of expected, and then maybe on that, what was found to be a surprise, like that you expected but then didn't find. Thanks.
Hi, Matthew. Yeah, so initially I used PIS data and IUCN distribution data to form my expected species list. Thereafter, Dr. Niels Kleinhans also evaluated my expected species list per region and per river system and all of that. But yeah, there is a few species that is on the list from all these sources which we didn't collect. Yeah, some I haven't barely identified. I don't even know how to identify from the names. But yeah, so there's a few, but we've tried to reduce it as much to museum and historically collected species in the systems and at sites.
Okay, thank you for that. I think we're getting close to the end of this really great session with very good discussions and questions. Is there any other questions? Wynand, I think you did mention there was more in the chat, or you said was it for a different session? Just any questions on YouTube, Wynand?
I'm just checking quickly. I think most of them was answered in the session. Yeah, no, I think there's nothing new that I can see now.
I saw Richard just ask a question to Annalise about whether you look at the flock list. Annalise?
Hi, yes. So I took that as well from the PIS data as well. So species with low frequency reference of occurrence, we kind of debated out based on other references if they will occur or should occur in the system, and then also on the available habitats and the stuff available from there for them to occur in the system. So yeah, historically, we looked at FPIS for maybe there's collection data of the different species.
Okay, thank you. Matthew, you've got your hand raised again.
Thanks, Nico. Just a question for Miss Ndluli regarding her study down in Nachel and in Underdam, and just the health assessment of Clarius charipinus. I know that we know that there's a subsistence kind of fisheries around these fish species in the area, but is there any maybe like recommendation that she can make in terms of whether these fish should be eaten or not in the catchment, and particularly the catchment like the Mgeni River itself?
Thank Thank you, Matthew. In terms of making a recommendation, I think the fish, Ashish has mentioned on the bioaccumulation, how the fish is accumulating metals. But based on the pollution around Nego and Inanda, I would suggest that fish from Inanda must not be eaten because of the level of pollution on that side. So the dam is receiving waste from 3 polluted tributaries, and it's also receiving waste from the land uses around Inanda. So I think the water quality there is bad, and the fish are not okay to be eaten.
Great, thank you for that. I think we've got about 3 or 4 minutes left for this discussion session. So the floor is open to any remaining questions to any of the 12 speakers within this session. So if there's any questions, I know if you look at the chat box, there was a nice question by Lazan, which he addressed to Lucille, and which Lucille answered as well. So just have a look there so that you can catch up on that. I see there's a hand by Gordon.
Yeah, I'd just like to add quickly, we've just found out that Mpumalanga Parks and Tourism is going to be moved to agriculture. And, we spent some time recently looking at the Water Act and how it really doesn't address biodiversity, where Environmental Affairs and NEMA does. So this is a broad question to the taxonomy group as well as the ecology group, is that what can we do with this information now? Maybe just a quick chat about how do we actually use this information to result in protection and conservation of those species on the ground? Because, we're not really considering biodiversity. Biodiversity's sort of been put onto the back burner as far as what we've seen on the ground here. Thank you.
Okay, that's a very broad open question. Is there anyone that's doing the diversity and taxonomy brave enough to answer that one?
Maybe I can just make a comment on that and link it to your comment or question to Albert earlier this morning, Nico, where you asked how does all this taxonomy and research on identifying new species and different lineages figure into assessment techniques like for Fry, and I think it comes down to what you want to protect and what some of these indices were developed for, where most of these indices were developed for water resource management rather than biodiversity protection as such. And when you apply them, what is the function, and you have to sort of ask yourself what's the function of a cryptic species that doesn't occur often in the ecological state of a system. And that's where it becomes tricky, because you don't want to lose species because you don't protect them, but you still want to get an assessment of what the current condition is based on species that occur more frequently probably than a cryptic species that occurs in very specific habitats, in terms of when you talk about water resource management. And when your focus is biodiversity, then the Fry is probably not your solution or your tool.
Great, thank you for those comments and views, Wynand. I think we've perfectly timed it. It's 12:15, and at least here in Potchefstroom. And so I'm going to close down this session. I would like to thank all the presenters for your presentations and also the discussions, everyone that asked questions and contributed to this really great discussion. As you know, the presentations will still be up for a while, so you can definitely go and have a look if there's ones that you've missed and now through the discussions has stimulated your interest. So please go and have a look at that, and then we can break for lunch, and then we're going to reconvene at 1:30 for the next exciting session in fish biology. So that's it from my side, and hope you have a good rest of the day.
Yeah, thank you, Nico. Just a reminder, please log in a few minutes before 1:30 when our next session will start, and then we'll enjoy your lunch and we'll see you after lunch.